All right. So hi everybody. Couple random things before we actually get into everything. First of all, I just want to show you these eggs. You'll see them again next week in your life. Probably on one people. Eggs, everybody. They've been hanging out in vinegar for like 2 weeks, so the eggshell part is gone, but the just rest of it's there, and they're like average egg size right now. We're gonna see them again next week, so I just want you to see them now so you know what you're looking for later. So eggs. The other thing is, oh, excellent. Today's class, we're finally onto cells, right? Like, the chemistry part is great, but we're finally onto cells. Out of curiosity, how is chemistry going for people? It's going okay? It's doing what? Ah. Okay, that's good. We're going to look at plant and animal cells, we're going to look at the nucleus, the ribosomes, the ER, and then just the whole endomembrane system in general. But before we do that, I actually have a couple questions for people. You got this. The first two are just administrative kind of questions. Currently, I realize you know this, but I'm going to so I'm going to explain to online people mostly. The homework, there's the homework follow-up, and there's also that reading assignment, the dynamic study thing. Got questions for that. Do you want it? I really don't care. The homework is staying. I love the homework. I think it's great. I also don't have to do it, so there's that. The reading assignments. It's probably helpful to learn stuff, right? Like, it probably forces some learning. If it were me, when things have hard points attached to them, I'm more likely to do them because I'm like, I've got other things to do with my life. So this is that option. So I'm telling your vote and also 9 o'clock classes votes. So this option is like, yeah, there were 5 points. It's not a huge deal, but if I do it, keep it for 5 points. This option is, yes, have it, but just have it be extra credit points, so you don't get points off if you don't do it, but you do get points for doing it. The problem that happens with this one is typically people who are doing great in the class do extra credit, people who are struggling don't, and it makes the divide worse. But then again, that's on you, whatever. And then the other option is to just have it for practice. So if you want to do it, you certainly can. It's good practice, but you don't have to. Any questions about this? Do you want to discuss or advocate for one or the other? No? Okay. I'm legitimately curious, because I was asking a buddy of mine about this, and they were saying, like, well, so don't people want 4 points? I'm like, I don't know, actually. Okay. Once yours reads, go ahead and put it down. So then I can get anybody who's not being read. Down. Did I miss anybody?
No.
Do it?
I don't like mine red.
Yeah, right?
They're good.
Did it? Anybody else? Anybody not have their paper thing? Do you have a vote? Are you okay telling people your vote? Actually, I'll see what it is. Well, okay. Yeah, 9 o'clock had similar vote. Okay, I can do that. Are people okay if this happens retroactively as well? So the first, yeah, I'm on it. Okay, the other thing, and I'm not meaning to shove all my decision-making process off to you all. In a normal semester, I'd have you all take the exam in class, and if you were like actively dying, you could take it in class, or you could take it like during my office hours or something else, right? This is not a normal year. There are going to be some people who are taking it online. Like, there will be some people taking it online. the problem with online tests, and I think I was yapping to you about this before, it's not a huge problem, it's just they have to be designed differently than in-class tests. So online tests typically need a time limit, because otherwise you just look everything up. And I know not everybody would, but there's some people I can't. Yeah, so there has to be a time limit, so it has to be kind of tight. So basically you read the question, answer it, read the question, answer it, read the question, answer it, as opposed to thinking about it. If you're in class, I can give longer time to be like, yeah, you know, consider, remember, reflect, ponder, right? Online questions also typically need to be more conceptual rather than which of these is X, right? And you can have both, but they have to be a little bit more that way. So the options for how we can do this test are, you can have everybody take it online. It's an online test, everybody takes it online, right? That's probably the most straightforward option. Kind of like quizzes, right? Just, you go, you do it. Does anybody know when we have this exam, by the way? Next Friday, yeah. That's that. So you wouldn't have class in person. You just, at the time, it'd be open for the hour. You take the exam then. The other option, well, one of the other options is we do this in class. So unless you have a doctor's note or the nurse's note or the I'm in isolation, quarantine, or sick, you come in class, you take it, you leave your paper on your lack of desk, and then the online people would have a different version. The other option is that weird hybrid thing of pick your favorite version. You understand what's going on here? Yeah. So, I'm curious. And again, I'd like to point out this continues to not be a democracy, but I actually do want to know what people prefer. That doesn't really make that much difference to me. I'll come back to you. Okay. Is yours the only one that's not reading? Oh, that's yours. Did you get you? Right, let's see where we're at. Everybody will be upset. Okay. That's the problem when there's three-way splits like this. We're like, well, no matter what I pick, I'll see what the more 9 o'clock class said. Okay, last one. This one is sneaky. I know it's sneaky. Okay? It's meant to be sneaky. We'll discuss afterwards. It's meant to, like, you know, encourage thought. Feel free to yap with your neighbor if you really want to. Long enough? People ready? Okay. Oh, right. It's supposed to show students. Huh. Sorry. Display while scanning. I think I'm scanning. I thought I was scanning. Alright. Here we go. Okay, great. Pretty sure I scanned 20 people like 3 times, but that's okay. I don't know what it did over there. Okay. Anybody else doing anything? Great. Ah. Gotcha. This one's right. This eukaryotic. Oh, good grief. Why is okay? This is a really good answer, right? Because the nucleus is the defining characteristic of eukaryotic cells, right? Prokaryotic cells do not have nucleus, right? Like they just don't. So this is a solid guess. This one's actually right because if you have any membrane-bound organelles, you have to be a eukaryotic cell. The eukaryotic cells all have nucleus as well, but presumably you just can't find it yet in your cell. We're okay with that? Okay. Apparently this was not as sneaky as I thought it was. I thought it was super sneaky. The next thing is a painfully adorable video. Like, it's painfully cute, and I recognize you're adults, but just bear with me, okay? We're not watching the whole thing. We're just gonna look at it. It's kind of disgustingly adorable. I realize this, okay? So just bear with me. I know they're kind of cute. But I also feel weird about showing them to you. We're gonna look at a couple things, then we're gonna see them all in more college form. Online people, this is going to be the YouTube link that's in the PowerPoints. It's not right now on the screen, but it's in the PowerPoints.
Now, to start our trip, we're going to have to get through this cell membrane, also called a plasma membrane. It's selectively permeable, which means that it'll only let certain select materials in and out. But by doing so, it keeps things in the cell stable, also known as keeping homeostasis. We have an entire video on just the membrane itself, which is found in all cells. But for now, we're just going to have to squeeze through this protein in the membrane. Now inside the cell, we find ourselves in this jelly-like material called cytoplasm. It surrounds all these internal cell structures, and you'll find it inside both prokaryotes and eukaryotes.
I know, right?
Now, organelles that are floating around in the cytoplasm can have more support than you might think. Cells contain a cytoskeleton, which is a collection of fibers that can provide support for the cell and its organelles. And the cytoskeleton can even play a major role in cell movement. The cytoskeleton actually deserves its own video because it's very complex and its organization can vary depending on what kind of cell you're looking at. Moving through the cytoplasm, let's start with ribosomes. They are not membrane-bound organelles, and they're going to be in both prokaryotes and eukaryotes. They make protein, which is really important because that's what so much of genetic material, DNA, codes for, protein. Ribosomes can be free in the cytoplasm, but they can be attached to another organelle too, which we'll talk about a little bit later. We're now going to focus on organelles that will be membrane-bound. So we're going to be focusing on organelles that you would find in a eukaryote cell. This takes our travel to the big boss, the nucleus. Now in eukaryotes, it holds the genetic material. Genetic material as in DNA, for example. Both prokaryotes and eukaryotes have DNA, but if you're a eukaryote, you have a nucleus to put it in. The nucleus controls the cell's activities, and inside it, it has a nucleolus, which is where ribosomes can be produced. Attached to the membrane of the nucleus, or nuclear membrane, you will find the endoplasmic reticulum, ER for short. It does a lot of processing of molecules for the cell, like protein folding, and it's highly involved in actually transporting those molecules around, like a highway. And there's a rough ER, which has ribosomes attached to it, making it, as you can imagine, rough, and then smooth ER, which doesn't have the ribosomes. Rough ER specifically tends to be involved with protein producing and transporting, because remember that ribosomes make protein. The molecules that leave the ER can be sent away in vesicles that actually pinch off the ER themselves. Now, smooth ER has many additional roles, including detoxification, which is one reason why your liver cells tend to have a lot of smooth ER. And another additional role of smooth ER is that it can make some types of lipids. Next, the Golgi apparatus. It's the ultimate packaging center. It can receive items from the transport vesicles that pinched off the ER. It has enzymes that can modify molecules it may receive and sorts the material it receives as well. It can determine where to send those molecules, including some that may eventually be sent to the membrane so they can be secreted, which means items that can be sent out of the cell. So with all that's going on in here, you might start to wonder, what's powering this thing?
Oh, I guess. No,
The
you don't.
mighty might.
Okay. It's them too. I was trying to stop before it got to that point. Okay. we're going to discuss all the things that showed up there. But just before we start that, we're talking about eukaryotic cells today. Just eukaryotic cells, okay? Prokaryotic cells are beautiful, but that's not what we're talking about. Eukaryotic cells have these membranes. They have to have these membranes. They're going to be internal, right? So all cells have this phospholipid bilayer on the outside. Eukaryotic cells have these membranes inside as well. Plants and animal cells have similar organelles. So this is an animal cell. It has lots going on here. They don't all have to have everything happening here, right? So you've got like a flagella, you've got microvilli. Not all animal cells have both or even either. You can see we've got the nucleus, we've got the ER, we've got smooth and rough versions, we have the Golgi, we've got ribosomes happening here, right? We've got mitochondria. Plant cells have the same deal. So they've also got ribosomes. Again, ribosomes are not organelles. They don't have that membrane. But they've got mitochondria, they've got the ER, they've got the nucleus, they've got the Golgi. Plant cells also get to have chloroplasts, right? And the big difference you can see here is plants have a cell wall, right? But they still have this plasma membrane. Just because they have a cell wall doesn't mean they don't need plasma membrane. They've got to have a plasma membrane. Okay with this? That was actually, we're going to move on. Okay. On to the chunks. DNA most DNA is going to be living in the nucleus. So there is going to be some DNA other places. The mitochondria is one of these places. Mitochondria kind of does its own thing. But most DNA is hanging out in the nucleus. And then ribosomes are what's going to use the information to make some proteins. Nucleus is going to be a storage center. It's usually the biggest organelle you can see. So when you stained your cheek cells in lab, whenever you did last week, did you guys do this, right? Yes? With a blue dye? No? Some of you did? Okay. My lab, I made you do it. Okay. Sorry, people in my lab, I'll address you. That dye stained the outside edge, it stained the phospholipid bilayer there. It also stained the phospholipids of the nucleus, right? So that's why you saw the blue outline with the blue circle in the middle. You were looking at the nucleus. It's going to be this bilayer. This is electron microscope, a scanning electron microscope, looking at the nuclear envelope. It's got two layers. We're right on the edge of both of them, so you can see one layer and then it's slightly more in focus here. It's got this layer as well, it's got two layers to it. All these little holes are called pores. They're going to be entry sites, so things can come in or out. Right? So for all the world, there's pores. So beautiful. For all the world, it kind of looks like a cell inside a cell almost. Right? It's phospholipid bilayer, some entry holes. That's the nucleus. You'll notice it's got the ER attached to it. The cell does not know, nobody has told the cell yet, that these things are color-coded differently, right? Like, the nucleus is connected to the rough ER. We call it different things because it's convenient, it has different functions to think of it differently. But it's kind of like if you're driving across the United States, other than the signs we put up, it's not inherently obvious when you leave Kansas and enter Colorado, right? Like, it's not like a big blue line on the ground. Cells also don't exactly know that there's a huge distinction between these, right? The membranes are going to be very, very similar. Here, this purple stuff that we're looking at, we'll see again in a minute. It's called chromatin. But this nuclear envelope, right, it's got pores in it, let things in or out. It's important. It's also got some structure to it, so the nucleus doesn't just mush together, it stays held open. It's held open by these proteins that line the nuclear side, the inside of the nuclear envelope. It kind of holds it tight. So far, so good? Feeling pretty comfortable with the nucleus? Yeah? Okay. Um, Anybody here like fishing or gone fishing before? Yeah. Okay. Maybe people are way more organized than my relatives, which is probably not surprising, but in your tackle box or whatever, you opened it up. Is there like this huge, terrible snarl of fishing line that, like, who knows why it's there? Nobody throws it away because it's good fishing line, but it's kind of a mess? Yeah, right? Because fishing line is really long, but it's also really thin, right? It just knots itself up terribly. Okay. DNA is very thin and very long. You do not want it to become that snarl fishing line. You don't want it to get tied up in weird knots, right? You want it to be able to be usable. So DNA gets organized in things called chromosomes. So a chromosome is one single molecule of DNA plus proteins. And the proteins, the DNA wraps around the proteins and it gets organized. We're good at this. We will see this again so much in like November. That DNA protein stuff is called chromatin, and then those get organized even more to become chromosomes. Everyone's with me there? Nucleus is holding genetic material in the form of DNA. This DNA is organized with proteins into chromatin, making chromosomes, and they're living there. The nucleolus is this glob right in the very center. It's where you build ribosomes. So you gotta have ribosomes to do anything, so the nucleus is gonna be able to build ribosomes. Okay, and now actually on to the ribosomes. The ribosomes couldn't be making proteins. That is what they do. They're called complexes, not organelles. Why don't they get to be an organelle? An organelle, they're complicated. They're not bound by membrane, yeah. So they're not bound by membrane. They're still complicated. They're called complex, but they're not an organelle. They've got two spots to do this. They can either be basically a freelance ribosome, which is just they're free in the cytoplasm. They make proteins just wherever it is they happen to be floating around. Or they can have like a steady job. Basically, they can be attached to the endoplasmic reticulum. Okay? Concerns about this? Oh, I should show you the picture first. This is the picture. This actually is another naming example that I'm okay with the naming. It's got a big part and a small part. It's called the large subunit and the small subunit. Just because whoever was naming stuff apparently got tired that day. Biology is really inconsistent about its naming things, because sometimes it's got things like large and small, and then other times it's got things like Sonic the Hedgehog protein. I kid you not. It's called Sonic the Hedgehog protein. I don't know why. I think it had something to do with whatever scientist was looking at it, probably had a small child who was like, Mommy or Daddy, next protein you find, can I name? And they said yes, because they made poor choices. It's abbreviated SHH, by the way. Anyways. Ribosome, large subunit, small subunit. You can see here. Nope. You can't. We'll look at it later. Large subunit, small subunit, ribosomes, they're going to be building proteins. Everything else today that we're going to talk about are part of what's called the endomembrane system, which is just saying basically it's stuff made out of membranes inside the cell. Lots of things to look at. Nuclear envelope we just saw, the endoplasmic reticulum, the Golgi, or the Golgi apparatus, you can call it that, lysosomes, vacuoles, and then the plasma membrane. We've seen the plasma membrane. We're going to spend all next week talking about the plasma membrane. All these little chunks either are physically connected or they're connected by vesicles, meaning transport happens between all of them. Okay, the ER, which is short for endoplasmic reticulum, because that's hard to say, right? It's got most of the membrane. It's got probably half in a given cell. Stuff is huge. It's got lots and lots of surface area. comes in two flavors of this. So we've got smooth ER. Can you see it? Is this big enough? Let me make it bigger. Over here, do you see this membrane? It's smooth, yeah? Online people right here, smooth. And then over here where you see this little blobs, I realize I'm saying this twice, but took me years. These little blobs, this is the rough part. What are these little blobs? Ribosomes, right? And so yeah, rough ER, smooth ER. The smooth ER is going to be making lipids. It's pretty good at that. It's going to be metabolizing a few carbohydrates. A big job for it is to be to handle some poisons or toxins in the cell, and it's also going to store some calcium ions. So it kind of has a series of functions. Making lipids is a pretty big one. Yeah, sorry. We're okay with that? Just kind of there. The rough ER is where a lot of things happen. It's got these ribosomes so many, and they're right there. These ribosomes that can read DNA to make protein, or smack up next to the nucleus, where they can read RNA, but genetic materials can come out of the nucleus and find ribosomes right there. So ribosomes are going to make proteins. They make proteins that typically aren't entirely finished. They're usually making something called glycoproteins. It's this protein. It's got a little sugar attached to it. The protein part is for proteins. The glyco part is for like glucose, right? This carbohydrate stuck to it. They're going to be making these proteins. They're going to be making transport vesicles. Any of this stuff is just churning out membrane as well. The Golgi apparatus, things go from, we'll see this whole picture in a minute, but from the ER, it's going to take stuff. It's going to take these little vesicles, these little packages that are full of proteins, full of glycoproteins. It's going to take these packages and pretty them up. So it's going to help get rid of maybe that glyco part, maybe it's, helps the protein be exactly how it's supposed to be. It's going to modify it a little bit, and then it's going to package it and ship it off to the rest of the cell. So far, so good? So the little random pictures here are supposed to be like to jog people's memories if it helps. With the nucleus kind of being the storage site, the basic informational part, right? Holding information. The ribosomes acting as like the machinery to build stuff. The ER acting as a factory. Did I not put? Yeah. The ER acting as like the factory that's holding these machines, and the Golgi is kind of the shipping distribution center. Yeah? And here's that picture. So here in the cell, there's a nucleus, ER. This Golgi apparatus is receiving vesicles, moving them through the Golgi, and then these vesicles are coming off. You can see it here in an actual image. Right, receiving vesicles, lots and lots and lots of membrane here for proteins and things to work its way through, and these vesicles are coming off. So membrane is leaving the Golgi all the time, right? This membrane is physically coming off, right? But this thing's not shrinking because it keeps getting membrane from the other side. Fair enough? Questions about this? Is this all old news, people? Or you're just absorbing it so quickly, you're like, yeah, got this. Okay. Next is my personal favorite organelle. I don't know why, I've just decided this is my personal favorite one. They're called lysosomes. Lysosomes are sacs, they're membrane sacs, they're an organelle. Membrane sac of what are called hydrolytic enzymes, which is basically enzymes of death and destruction, right? It's basically this bag of stuff that if these enzymes come in contact with things, they will tear it to pieces. So this seems like a terrible, terrible idea, right? Like, you wouldn't probably like if someone handed you a bag full of, hey, this is a bag full of, like, face-melting enzymes. Please hold it carefully, right? Why on earth would a cell... Why do they have these? Get rid of the bad stuff. Yeah. Has anybody here ever had, like, a seriously crappy couch? Like, it's terrible. And then you're like, I should upgrade my seriously crappy couch to a slightly less crappy couch. And so you post this on Craigslist. You're like, beautiful couch, free to a good home. You just pick it up, right? And nobody picks it up. This has happened to me before. Nobody picks this couch up. You're like, huh. So then you shove it out to the street with a sign, free to good home. And nobody takes it. You're like, how do I get rid of this thing? Right? Cells have a similar problem. So if there's something, let's say there's an organelle. Let's say there's a mitochondria, right? It's old, it's worn out, it stopped working. It's taking up a lot of space in the cell. It's not doing anything in the cell. And there's a lot of, like, good stuff. There's a lot of proteins. There's lots of minerals tied up in this thing. You want to get rid of it. You want something like a lysosome to come and chew it to bits so you actually can do stuff. Are we okay with that idea? There's actually, it's sad, I think happy stories. I'm trying to think of happy stories, sorry. There's a genetic disorder, it's called Tay-Sachs. You heard about it? Yeah. It's a problem with lysosomes. So lysosomes don't do their job, and cellular wastes just accumulate in cells, and it's a terrifying genetic disorder because the parents, and the kids probably don't know yet, but the parents know it's just a slow burn, right? Like, it's good, the cells are filling up with waste, and it's rough. But typically, lysosomes do a really good job of tearing things into bits. Lysosomes can break down their own things. It's called autophagy. It's self-destroying, right? Or they can break up things like food, little particles of food. So let's say the membrane takes in some particle of some, like, big protein or something. The lysosome can fuse with this, open it up, and then you've got the enzymes here that can break this apart. So far, so good? You don't have these enzymes just floating around the entire cell. You have it in a controlled way. Concerns? Okay. Oh, and here's that picture here. Lysosome. So I've been using the phrase vesicle a bunch of times now. A vesicle, are we okay with this just being a small membrane sac, basically a little bag of something? If you've got a larger bag of something that doesn't exactly fit it's not the nucleus, it's not the Golgi, it's not the ER, it's not a lysosome. You can call it vacuoles. It's basically the miscellaneous category. It's big enough to be something special, but it's the miscellaneous category. They're basically really, really large vesicles. They're coming from the ER and the Golgi, right? Really large vesicles, and they're doing something. So we just saw one here, this little food one, right? The food ones, sometimes certain freshwater protists have, they're called contractile vacuoles, and they're pumping out water out of the cell. Plants have a really, really big one. Can you see it right here, this really, really big structure? Plant cells will often hold water in this thing. You're not expecting this in an animal cell, but a plant cell, it can hold water beautifully well, or it can give off water, and they're often quite large. And they can do a bunch of different functions. So far, so good? Oh, okay. I did show the other class another video, but I hustled through more with this one because last class I was pushing it on time, and this one like, let's just move, people. I should probably, no, stop computer. Sorry. Great, we're gonna do some thought questions at the end here because we are way faster than the other class. Sorry, I think I stopped recording. Just a moment. Nope, I'm still recording. Okay. This is the picture of the overall where everything is, right? So this whole thing is the endomembrane system. So help me out. What part is actively like making lipids themselves? The smooth ER, right? Which is a really nice organization for this because the smooth ER can be making lipids. And hey, what's making membranes? So smooth ER is making lipids. What's actually physically making membranes? Is it the nucleus? Not really, right? Nucleus is going to be holding on to some genetic material and putting out some genetic material, but it's not actively. This nuclear envelope here, you're not expecting it to get any bigger. You're not expecting it to get any smaller. It's just kind of doing its own thing. Okay, smooth ER is making lipids. Rough ER will actually make membranes, but has another job too. What's the other job of the rough ER? To hold these ribosomes to make tons of proteins, right? Churning out protein. You can make protein other places in the cell. You can have 3 ribosomes making proteins other places in the cell. But this one, there's just so many of these and they're right next to where they're getting material. It's gonna be great. So making membranes, making proteins, little bits will glob off in this vesicle, will come off. But this isn't going to get smaller because this is actually making membranes, right? This vesicle will come and fuse. There are sides to the Golgi. There's what's called the fifth side that's close to the ER, and the trans side is far away from the ER. Okay, so vesicles typically are coming in to the cyst side, moving through and going out the trans side. In certain scenarios, you can run it backwards, but for us, this is the direction of it. So far, so good? What's happening in the Golgi? Like, if you have a vesicle coming here and then coming right out, why not skip this whole middle step and just go from here to wherever it is that you're going? Seems like an extra endomembrane portion. It can modify it, right? It can do stuff to it, which is good. Like, it can modify it, and then it can also pack these into like a whole bag full of this stuff. But yeah, it gets shined up. It's folded nicely by the time it ends here. It's got typically whatever glycobit needs to be off is off. It's great. And then vesicles can leave as lysosomes or as just miscellaneous other compartments, or they can go all the way to the plasma membrane, and you can get the proteins out. So for instance, like if you have some sort of hormone that you're producing, let's say insulin, right? Your cell will happily make it. It'll package it in this vesicle, bring it to the membrane, open it up, and dump it out. We'll see membranes more later, but this protein going out, is that making a hole? Is that making a physical hole in this membrane? No, I do have a video to show you. It was up before, but then the power killed it. Let's see. Which one? This one. You don't need to worry about the snare. I'm just going to show you the picture. I'll turn off the sound, actually. So it's got a bunch of endomembrane parts coming off, moving through, everything's great. Oh, question. What's this supposed to be? roughier, this Golgi, yeah. Buses are coming through. The similar pattern is going to happen if it hits the outer membrane too. Phospholipid bilayer here, we have vesicle, phospholipid bilayer here, right? Neither of them have big holes in them yet. Two different phospholipids. Also, online people, I'll put this link will be in the lecture notes as well. Good grief. Come on. And it gets pulled together. The mechanism, don't worry about the mechanism, but it's these two membranes are going to get smooshed together, and they're going to fuse. So now the membrane's bigger. The proteins have made it to the other side, and there are no holes anywhere in this. All right. This is how you move proteins around between things. This mechanism also works if you want to make the cell membrane itself bigger. So like if you're a neuron and you want to grow those little sticky out bits like the dendrites, if you want to grow those bigger and you need more membrane, you just bring a vesicle, you bring membrane to the edge, have it fuse and open it up bigger, right? You can also run this in reverse. You can pinch bits off and pull it in. And that's next week's project. Any questions about this? Any questions on things that are coming up? The quiz is Friday, this Friday. So next class we have a quiz, and then a week from then we don't have a quiz, but do have an exam. Yeah, it's like the, I don't know, like the 18th or something. Is that right? Okay. Sweet. That's what we've got. And sorry, I go through this way too fast. Are you sure you don't have questions? Huh? Either you guys are more awake than the 9 o'clock class, or I talk.